chance to get your sample, you will have time later. Before we get underway, I want to talk about a few things, just wrapping up the end of the semester. So the first half today, we're going to do the urinalysis material for the urinary system, and in doing that, I'm going to talk some about aspects of the urinary system that I want to cover. And then the second half today, we'll deal with the reproductive system. And it's not really enough time to do what I normally do for the reproductive system. So I'll truncate that a little bit and highlight the stuff I want you to pay the most attention to and let you do some of the review of the anatomy, the basic stuff there. I'll just cover the tougher things today. And then next week is our last meeting, and we'll be doing the second practical. So let me go into that folder for a second and just talk about a few things. I think it was, well, no, I guess it was a few weeks ago when I was talking about the previous practical, and I thought about making a different type of study sheet. I didn't do that. It's been kind of a crazy week. A lot of the time I had available, I got caught up on your grading. So it's your fault for doing the work in the class that I have to grade. And I didn't get to do stuff. So I still have the same outline and stuff as before. So there's cases and then the online practical assignment. Also in this folder is the cumulative final assignment, and I just want to talk about that for a little bit. I used to have students do the second practical and the cumulative final both in class on the last day, and the final exam period for the regular semester is 1 hour and 50 minutes. And so they'd have to basically do 2 tests in that time. They're both 50 multiple choice question tests. You're familiar with the practical. It's exactly the same as the first one. And then the final is also 50 multiple choice questions. And so really a 50 multiple choice question test can be completed in about 1 hour. So it was really cramming things into that just shy of 2 hour time frame. And what often happened is people would spend 1.5 hours on the practical and then they don't have a half hour on the final. So I switched to doing the final online, which made it possible for me to do a few things with it that I'm actually kind of happy worked out. The paper version, there were 50 set questions that everybody got. And with the online version, the questions are actually drawing from the same pools as the work that you've been doing all semester. All of the part 1 and 2 assignments for each chapter are just reused here. So you're going to be getting questions you've already seen. Like I said, the test is 50 multiple choice questions, and I think there's like 10 units worth of material. So roughly about 5 questions per chapter. And that's pulling from the pool for the 2 assignments. Pools range from 20 to 40 questions apiece. It depends on how many I had written, and I add some every so often. So for one chapter, there's 2 pools, which can be 40 to 80 questions, and it's just pulling 5 questions out of those pools for that chapter. You won't know necessarily which questions you get. Obviously, it's randomly drawing them from the pool. And the chances of 2 iterations of that test being exactly the same is practically nil. The chances that the same 50 questions are pulled from all those pools for 2 different people at the same time is pretty slim. And then on top of that, every time a new question is pulled out of one of those pools, if you haven't noticed it yet, The A, B, C, and D choices are changed randomly. For most, there's a few where they stay the same. It's usually when there's numerical answers, and I put them usually in a particular order. Those don't change, but the vast majority do change, which is the 4 choices, what order the 4 choices are in. So the chances of 2 people getting the exact same test is slim to none. Preparing for it is, before I get there, it is a multiple choice question test, and the other assignments you've had like that are timed. This one is also. I've been timing these based on 2 minutes per question. So for 50 questions, that's 100 minutes, or an hour and 40 minutes, which is more than enough time to complete something like this. Like I said, it used to be people would complete it within a half hour to an hour in class after they finished the practical. Of course, they rush through about a bit of it, I'm sure, but an hour and 40 minutes is plenty of time for this kind of assignment. You have 3 attempts to complete it. Those 3 attempts will not be identical. You're actually not likely to have any of the same questions on those attempts. Again, because it's drawing from such a large pool. So you can expect it'll be a little bit different there. The best of the 3 scores is the one that goes in the grade book, not the last, but the best of the 3 scores. I started to say it and then I moved on and talked about the time thing. But let me go back to it. Preparing for this is straightforward. I wouldn't say easy because it doesn't include some effort, but all of the questions that'll be on the test are available to you to look at beforehand. It is a timed assignment, so you don't want to pull up the assignment and then go back and look for the questions that are on it. Instead, you can go back and look at the questions to prepare for it so you know everything that's going to be on it. And all you need to do is go back to the part 1 and 2 assignments that you've been having all semester. If you did not take those assignments enough time to see all of the questions in them, you can go back and reopen them, and the assignment will pull more questions out of the pool, and you'll be able to see more of the questions on the test. The answer choices will, of course, be in a different order, so you can't say, oh, this question, the answer is B, but you can at least understand what the answer to each question is, and so if you always sit down and take this, you should be completely prepared to just go ahead and breeze right through it. Remember that the last attempt that you take on any part 1 or 2 assignment is the one that counts. So if you go back in to look at these assignments, to look at the other questions, be sure that you finish with a strong answer, a strong score. And really, you should be able to get all of those questions correct because you're studying those questions to be prepared for the cumulative final. You can go back and redo any part 1, 2, or 3 assignment, although the part 3 assignments have nothing to do with the final. But you can go back and do those, anything from the whole semester, regardless of due date. And I would strongly suggest you do that to bring your score up to 100%, which is just going to help your grade in the end. If you're doing that, whether it's to bring your score up or to prepare for this, I strongly suggest that you look at those assignments in reverse order. If you have a part 3 assignment that you've done before and you finished that chapter, you did part 4 for that chapter and everything, but you want to go back in because you got 16 instead of 20 points or whatever, start with a part 3 assignment, because when you open up one of these assignments after the due date, it will remove the link to whatever assignments are dependent on that. So if you open up a part 3 assignment, the link to part 4 will disappear. Any work you've already done on part 4 is not lost, just your access to the assignment, which doesn't matter for part 4 because you've already done the part 4 assignments. And you're just going back into part 3 to bring your score up. Then go to part 2, bring that score up, make sure you look at all the questions to prepare for the final. because it's randomly pulling questions, I can't say for certain, but probably 5 or 6, maybe 8 attempts on an assignment, you'll see all of the questions. It'll be obvious when you start pulling up assignments, you don't see any new questions, you can assume you've seen all of them. So do the part 2 one. When you open the part 2 to do that, then the part 3 link will disappear. Okay? So then if you're working on part 2, you can't get into part 3 to improve on that. And then when you're comfortable with the part 2, you've got the 100% score and you've studied it to prepare for the final, then go into part 1. Once you open part 1, then the link to part 2 will disappear. So if you started with the part 1 assignment that you've already done, you won't be able to get into part 2. And if you can't get into part 2, oh no, that's not true. If you've already done them, you can't get into part 2, you just can't get into part 2. You can still get into part 3. But for studying for the cumulative final, you want to be able to get into parts 1 and 2 to look at all those multiple choice questions. Actually, it just occurred to me, I want to change this. Last semester, for those of you that have me for MP1, I started this, which is, I'm just clicking this button here, which is to say, after 100 minutes, the test will auto-submit, okay? All of the other timed assignments you've had, if you go over the time limit, it just says time's up and it doesn't kick you out. But this will, in fact, kick you out. You should be able to complete it in 100 minutes because you can prepare for it by looking at all the previous questions. And really, you should sit down and as long as it takes you to click 50 little buttons, you should be able to finish the test. So that's plenty of time. If you hit the time limit, it'll kick you out. Any questions you haven't done at that time will just be left blank and be calculated into your score. That was a new policy I started with my classes last semester, which would affect any of you that had me for MP1. I didn't have that policy in effect last summer, so I had to go ahead and change that because it wasn't in the system there, but now it is. That's the deal with the cumulative finals. Now the due date for the cumulative final and for the practical assignment there is August 6th at 11:59. Okay. All of the other assignments that you've had have been due before you come in for the next class. This one's due Thursday of next week by midnight. After Thursday, I'm going to get onto Blackboard and shut down your access to that. I might do it at midnight when it switches over to August 7th. I might do it in the morning when I wake up and take out the dogs. Kind of depends on whether I'm awake at midnight. So some semesters I do that right away. Sometimes it's in the morning. There's no telling what it's going to be. So be prepared to complete the work by midnight. And that goes for all of the work in Blackboard, even stuff that's past due at this point. If you haven't completed it, you have to complete it. Says right there. Right, I know. But when you go into this assignment, you'll see it say specifically the cumulative final and practical have the ultimate due date set for them. So all of the work on Blackboard that you might not have completed or that you want to work on some more to bring your grade up has to be done by midnight on the 6th. Anything that you have not completed will count as a 0 in the grade. Right now, it's not. The way that the grade book works in Blackboard, I have it set up so that it only calculates your grade based on work that you've completed and that's been graded. Now, the auto-corrected stuff, parts 1, 2, and 3, and lab quizzes and that sort of thing, as soon as you complete it, the grade's there, unless you're doing it late. For the part 4 stuff that I have to grade by hand, those don't get calculated into your grade until I've done the grading. But anything that you haven't done yet is not factored into the grade, because if it were, you'd have an F all semester until you completed everything. So it's showing you how you've been doing all along based on the work that you've completed. If you did only one assignment at this point in the semester, one, you're screwed, but two, that would be your whole grade. Okay, so if you did one part one assignment, got 10 questions correct out of 10, you'd have 100, which would be an A for the lecture part. And all of the other work that you hadn't done yet would be calculated in. So you'd be going, oh, look, I've got an A, I'm gonna ace this course. But you have a lot of work to do in the meantime. And if you, in fact, are in that situation where you haven't done most of the work for the course, in my experience, you're not going to do very well in this class. I say at the beginning of the semester that you really should stay on top of the due dates. They are a little fluid because things come up. But if you've been taking that to mean, oh, hell, I don't have to do it until I want to, your day of reckoning is coming very quickly. If you are in that boat and you have work to do, except for the practical and cumulative final, everything is past due. Okay. Well, no, that's not true. The stuff that we're covering today is due next week. But everything else is past due. So if you're doing work now from June, you're going to run into the it being marked late. You do a part one assignment, I have to check it off before you can do part two. And then you do part two, and I have to check it off before you do part three. I do not get on Blackboard every second to make sure that everybody's up to date. I might look at it once a day. Actually, this summer's been kind of crazy, so not even that often. You can contact me by text or email, say that you're waiting on me to clear off some late assignments, and I'll get on as soon as I can to take care of that. But the turnaround time to get through an entire chapter's worth of stuff, part one, part two, part three, and part four, if you have to tell me each time that you're waiting to get things checked off, is more than 24 hours. Okay? Don't anticipate that I'm going to be able to get back to you right away and unlock stuff that you're waiting on. So if on the 5th you say, oh shit, I got a bunch of stuff to do, you're going to be in a really bad situation. Try to maximize things. If you have several chapters worth of stuff to do all of the part ones, and then when I unlock all that stuff, do all the part twos. Start doing that now, you'll have everything done by the end of this summer semester. So. Any questions about the assignments that need to be finished up? So yeah, the online practical thing, just like last time, 4 extra questions per case. And that needs to be done by the 6th also. Actually, historically, the second practical is the last thing that we do. And so when I used to do the online part as more of a take-home paper test, I'd hand it out the last day of the class, and then a week later people would hand it in when they came to take their practical. So actually, practical 2 assumes that you've looked at the online questions already. There is actually a question or two in there that's sort of about what you would have covered going through the questions in the online practical. So if you're going to put off doing that until the 6th, at least look at it before the test, because some of it is relevant to completing the test. Any of the online practicals that are done when you come in next week to take the test, I'll be grading them most likely while you're taking the test. So so those are the assignments for the end of finishing up the semester. Next week, all we're doing is in the class portion of Practical 2. Since that's all we're doing, we don't need the whole period to do that. We don't need 4 hours. You get an hour and 50 minutes, again, based on the traditional, regular kind of semester final exam schedule. Which means there's no point for us being here at 8 o'clock. We'll go ahead and start next week at 9 o'clock, which will give me time to set things up and whatnot. You'll still be done by 11 o'clock because an hour and 50 minutes after 9 o'clock is obviously 10:50. So you'll be out of here by 11, possibly earlier since it depends on how long it takes you to complete that. Some people will be done by 10 probably. But you'll have until 10:50, 11 or so to finish things up. Now, I do want to mention saying that the rest of the campus is not putting off starting that day for an hour, so parking might be harder at 9 o'clock than 8 o'clock. If you have trouble finding parking, normally come on in at 8 o'clock, get a good parking place, and wait out till 9 o'clock, but we won't start until 9 o'clock. If you are fine parking wherever you can get a spot, and with all the construction and stuff, I don't really understand how parking's working right now. Yeah. But if you're okay coming in at 9 o'clock and getting a parking space, that's fine. Just understand if you come in for 9 o'clock and you have a hard time finding a parking place, you don't get here till 9:15, you'll still only have till 10:50 to complete the test. Okay. So that's for next week, starting at 9. I'll have the window covered up there, and I'll open the door at 9 o'clock when I'm ready for everybody to come in and take the test. And you'll, of course, luxuriate out on the hard floor outside. So, any questions about next week? Okay. Any questions? Yes.
The overview.
The overview. What about the overview?
So there's one in week 8, I think it was.
Oh yeah. For the environmental exchange chapters, the respiratory, digestive, and urinary system chapters, there's an additional assignment that ties those 3 chapters together. Actually, let me go to the thing. So yeah, Unit 8, which is the urinary system, which we're doing hopefully soon. There's actually an additional assignment here called Environmental Exchange Overview. You don't see the link to this assignment, but I did put a little announcement there saying that there is an overview assignment. There actually was supposed to be another one earlier in the semester, but I forgot that it's not in the summer course, it's in my fall course. So I forgot to put it over on time. But yeah, so I have this. You have to complete urinary system part 4 to be able to do this. There's no score requirement like with the part 1s, 2, and 3. You don't have to get 80% correct on this to go on to this. You just have to complete it. Also, since it's not score dependent, if for some reason you're doing that part 4 assignment after its due date, which is next Tuesday, you don't have to wait for me to check that off to go on to the environmental exchange overview. That's just another like part 4 assignment. It's 3 open-ended questions worth 6 points apiece for a total of 18. So it's exactly like a part 4 assignment. It's just questions about how the respiratory, digestive, and urinary systems relate to each other, okay, which is one of the themes of this portion of the course. So. Any other questions? Okay. So I did mention that I got caught up on grading. I got caught up as of Monday afternoon. I didn't get to do anything more than, I think yesterday afternoon I might have gone through and checked off auto-corrected things that were late, but I didn't grade any more part 4 assignments that might have been submitted. But still, you probably have a pretty good idea of where your grade stands at this point. Just understand that it's a little bit misleading because, again, it doesn't take into account anything you haven't completed yet or hasn't been graded yet. I will probably get 1 or 2 emails sometime in mid-August saying, how did I get this grade? I had a such and such on Blackboard. When I shut down access to Blackboard, then I go ahead and grade everything. I switch the grade book from ignoring incomplete material to counting that. And that often makes a difference in the grade. Okay, so if there's anything that you haven't completed and you're looking at your grade and you're like, oh, I've got an A, I'm okay. and you forgot to do something, as soon as that 0 gets factored in, it's going to bring your grade down. So you need to be sure that you complete everything. Go into My Grades. It lists all of the assignments. And if there's a dash where the score should be, that means that you haven't done that assignment yet. If there's a blue icon, I can't remember quite what it looks like. I think it's supposed to look like a clock, like you're in progress. That means you've opened the assignment, but you haven't submitted it. Anything that's in progress, when I go through, I will submit it. I have the power to submit your assignments that you haven't submitted. Whatever state it's in. So if you open it up but didn't complete it, and I submit it, then it'll be a 0. Okay? Or if you open it up and did one question but didn't do the other 2, if it's a Part 4 assignment, I'll submit it and you'll get up to 6 points on the one question you did instead of the up to 18 on all 3. So look at anything that's in progress, because you might not have completed it, and that can affect your grade too. Question?
For the makeup assignments, when they're due, is it the gray circle or would it be blue?
The gray circle means that you're exempt. If you hold the mouse over any color icon, circle, whatever, a little word will pop up telling you what that means. So if it's over the blue one, it'll say in progress. If it's over the green one, it'll say needs grading. And if it's over the gray one, it'll say exempt. Okay. So if you see a gray circle, and you can verify this by holding the mouse over, it'll tell you that you're exempt. If you are not exempted for something that you are sure you should be, feel free to contact me. Mistakes sometimes get made in doing that. So I can just double check the attendance and make sure. And you might feel that you shouldn't be exempt. I mean, you should be exempt from something, and I'll write back saying I have you down as being late on that particular day. That'll be the reason that you're not exempt from it. So, but yeah, go through and make sure that you complete everything. If you feel that you should be exempt from something and you're not, and you don't say anything, you will not be exempted from it because of that. It'll be counted as a 0. Okay. So look through the My Grade section and be sure that you have everything completed, and any of the makeup assignments that you feel that you should be exempted from. Check with me and I'll tell you whether you are in fact exempt or not, because part of being exempt is being here and on time. If you're late, which happens for several people on a fairly regular basis, you might be required to do makeup assignments for days that you were mostly here, but that still counts. So, I think that's everything. Any other questions? Okay, so let's actually get started on some stuff. Part of what we're doing this morning doesn't really lend itself to being recorded, so I'm going to just sort of record the preamble explanation stuff before we get into actually doing the urinalysis. And then I'll stop the recording. So if you're not here in this room right now and you're listening to this, it's going to stop and you're not going to get everything that I cover. You should be coming to class on a regular basis. Okay. So let me just go into the urinary system stuff. Okay. So the chapter starts with a section on the physical characteristics of urine, which is to some degree really looking at some of the things that urinalysis is about. So I'll put this up on the screen, so there's something on the screen, but I'm actually going to be talking a little bit about things with some models I have up here. Now first I just want to talk about, and I'm not going to put things up on the screen to go with this for the recording because it's just too much of a hassle for me to jump back and forth between the models and the computer view. So the structures that make up the urinary system, it's a fairly simple list. There's the 2 kidneys, the 2 ureters, which are the tubes that connect the kidneys to the bladder, of which there is 1. And then there's the urethra, which is, there's also only 1 of those. The kidneys are for filtering waste products out of the blood. I'll be going into a bit more detail about that, setting up what urinalysis is for. The ureters are just for conducting or conveying that urine down to the bladder. The bladder stores that, and then the urethra is for conveying the urine out of the body. The bladder is lined with a mucosa that has wrinkles in it, just like the stomach. They're called rugae. I introduced that term in terms of the stomach, which allows for the mucosa to stretch out a little bit. When it's empty, the rugae are present. When it starts to stretch out, the rugae flatten out, allowing for the mucosa to expand a little bit. The bladder can actually expand more relative to its empty size than the stomach can. And the reason for that is because the bladder has a specialized epithelium lining. What do we call the epithelium that's special for the bladder?
Transitional?
Yeah, transitional. Which is sort of referring to the fact that the cells transition in shape and size. In a relaxed bladder, the transitional cells are usually big kind of pillowy round cells, and then they can be stretched out and they become thinner bladder. We wouldn't call them squamous. They're still transitional cells. They've just been stretched out a little bit. It allows for the bladder to stretch out and accommodate more volume. Transitional epithelium is also found in the ureters. They're also there to help store a little bit of urine. If your bladder's full and you don't have a chance to empty it to avoid your bladder, some of that urine can back up into the ureters, and the ureters can expand also to accommodate extra volume. You do not want the urine to back up all the way to the kidneys. If the urine gets in the kidneys don't function properly, which I believe I said last week. Looking inside the bladder, you can also see this triangle structure in it. The two corners off to the sides are where the ureters are attached, and the corner in the middle is where the urethra is. It's called the trigone. It basically represents the three major attachment points for the bladder. It also acts as a funnel sort of system to direct the urine towards the urethra when the bladder contracts. The bladder contracts because it has a muscular wall, smooth muscle wall, just like most of your other organs. The muscle of the bladder is referred to as the detrusor muscle. I think I pointed that out last time. I talked about the holding it and the voiding reflexes. So the parasympathetic system will cause the detrusor muscle to contract to void the bladder. The sympathetic system will actually encourage it to stay relaxed as you're storing urine. Indistinguishable under the microscope from the detrusor muscle, there's more smooth muscle around the opening to the urethra, which is the internal urethral sphincter. It is controlled opposite to the detrusor muscle. It contracts under sympathetic control, and it relaxes under parasympathetic control to coordinate with the detrusor muscle that causes voiding. Students will often look at this model and think that they're looking at the female urinary system because there's such a short urethra. But in fact, the female urethra would be about twice as long as what we see in this model. And there's a very big clue that we're looking at the male system. This purple thing around the urethra at the base of the bladder is the prostate gland. Women do not have a prostate gland. The urethra for the male reproductive system would then, of course, be much longer because it's also part of the male reproductive tract. The two tubes that we see sitting sort of in the superior lateral position, I guess we call this, off top side of the bladder here. These represent the spermatic cord, which contains the ductus deferens that sperm travel through. I'm going to be pointing this out later on. It connects to the seminal vesicles, which we see on the posterior bladder here, and the sperm traveling through the ductus deferens and fluids from the seminal vesicles go into the prostate gland and then eventually make their way to the urethra for the male reproductive tract. So we see those structures in this model here too. So I want to talk about the kidneys in more detail. Before I switch from this model to the other one with bigger components, I do want to point out how this model represents one of the kidneys. The kidneys are made up essentially of two major regions of tissue. There's the outer tissue, which tends to be colored lighter in models like this, called the cortex, the renal cortex. And then the darker, more centralized tissue is the medulla. Now, usually in organs that have a cortex and a medulla, like the adrenal gland that we looked at in the microscope for the endocrine system, or the ovaries, which I'll mention later on, the medulla is just the center mass of the organ, and then the cortex the outer covering. For the kidneys, that's not exactly the case. The medulla is actually organized into discrete little clusters of tissue called medullary pyramids. In 2 dimensions, when we cut through them, like the bottom half of this model of kidney, they have a kind of triangular appearance. So 2 dimensions, triangle, 3 dimensions, pyramid, where the word comes from. They're actually not really pyramid shaped, they're more conical. They look to me more like popcorn kernels or something like that, unpopped popcorn kernels, which the top half of this model represents. The cortex has been cut away as if it's been cut in the frontal plane, but 3 of the medullary pyramids are left intact in kind of a 3-dimensional shape, so you can see what they look like. The medullary pyramids are connected to an empty space in the middle of the bladder, middle of the kidney, where the urine starts to collect before it moves down to the bladder. Each pyramid is attached to a structure called a minor calyx. Calyx is C-A-L-Y-X. In this model, we can see one minor calyx kind of sticking out by itself here. There would be another medullary pyramid attached to that, sitting in front of everything else on this model. So they don't show the pyramid, they just show the minor calyx that it would be attached to. And we can see some other minor calyces attached to that. Calyx is singular, calyces is plural. That's C-A-L-Y-C-E-S. Minor calyces come together in groups of a few as a major calyx, and then all of the major calyces come together at the center into what's called the renal pelvis, which is just the empty space in the middle of the kidney. The ureter comes off the renal pelvis, and the urine drains off of that. So I was pointing that out with this smaller model of kidney because I like the way it's represented, but let me switch to this model here, which has a larger representation of the kidney. It does not have the medullary pyramids represented in 3-dimensional shape like the other one. They're all cut in a sort of frontal section through the kidney, but we can see the minor calyces at the tips of the pyramids. Actually, the tip of the pyramid is called the papilla, which means little bump. So the papilla opens up into the minor calyx. And here at the bottom, we can see 2 minor calyces coming off of these 2 pyramids, and another one suggested out of the plane of section. Those coming together into a major calyx, and then all of that coming together where the renal pelvis would be, which is the part of the model that has the number 500 written on it, and then the ureter coming off of that. The kidney's job is to filter the blood so we can see the blood supply coming in. The book runs through the components that make up the blood supply. Let me just run through them quickly. I'm going to go through them probably faster than you can write things down. So go to the book for that. Blood comes in through the renal artery. It branches into what are called segmental arteries going to different regions of the kidney. And then those segmental arteries branch into interloper arteries, which go in between the lobes of the kidney. One lobe of the kidney is a medullary pyramid and its surrounding cortex. So the artery that we see going between these 2 medullary pyramids is an interlobar artery. The interlobar arteries branch and arc over the top of the medullary pyramid, called arcuate arteries, representing their arcing pattern. And then branching off the arcuate arteries are the cortical radiate arteries, which radiate out into the cortex, hence the name. The book calls the cortical radiate arteries, I think, the interlobular arteries. I hate that name because it's easy to confuse with the interlobar arteries, and it's not nearly as descriptive as an anatomical term. So I will continue to call them the cortical radiate arteries. I think there is a couple places in the book where they mention radiate or something like that, but interlobular is just another name for cortical radiate. Then coming off the cortical radiate arteries, which we can see here in this pie-shaped piece of the model, are arterioles, which then go to a little unusual capillary bed called the glomerulus, which I'll talk about later on. And then coming from the glomerulus is another arteriole that goes out, which we see best over here. And that gives rise to a capillary bed called the peritubular capillaries. And then coming off the peritubular capillaries are venules that merge into the cortical radiate vein. Cortical radiate vein goes into the arcuate vein, which travels parallel to the arcuate artery at the border of the medullary pyramid. And then that feeds into an interlobar vein, which goes between medullary pyramids. Bless you. For some unknown reason, there is not a name for what is obviously a different structure here. The interlobar veins continue all the way down by name until they reach the renal vein. Okay. There's this artery right here that I'm pointing at is the segmental artery. The vein that runs parallel to it is still called the interlobar vein. Don't know why that little pair got skipped, but otherwise, you have the renal artery and the renal vein, interlobar arteries, interlobar veins, arcuate arteries, arcuate veins, cortical radiate arteries, cortical radiate veins. The names represent the mirrored or parallel blood flow in and out. Once we get to the cortical radiate vessels, things get unique there. So we have the arterioles coming off, going into the glomerulus, and then going from the glomerulus to the peritubular capillaries, and then the venules going into the cortical radiate veins. So all of the stuff up here in the cortex is unique, but feeding into that and then draining off of that is the mirrored process of the arteries and veins, except for the segmental artery. There's not a comparable segmental vein. Although, if you look at it, there is obviously a vessel right there that looks like it, but it's still called the interlobar vein. Now, the glomerulus, this funny little capillary structure here that has an arteriole going into it and an arteriole coming out of it, is connecting the blood supply to this tubular structure that we see here, which is called the nephron. The nephron starts with the capsule that encloses the glomerulus, which is represented in the third part of this model, and then tubes coming off of that. The tube coming off of the glomerular capsule to begin with is called the proximal convoluted tubule. Proximal because it's right next to the glomerulus, or more accurately, it's connected to the glomerulus. Convoluted because it has a bunch of twists and turns in it, and then tubule because it's a little tube. The proximal convoluted tubule then gives rise to the descending limb of the nephron loop. Some sources will call this the loop of Henle, but I prefer the non-dead white guy name, just calling it the nephron loop. So there's a descending limb that goes down into the medullary pyramid, and the ascending loop that comes back up. At the top of the ascending loop, the nephron then gives rise to the distal convoluted tubules. Distal because it's far away from the glomerulus in terms of the flow. Convoluted again because it's got twists and turns to it, and that's a little tubule. In fact, the proximal and distal convoluted tubules are both
situated next to the glomerulus. It's just that the proximal convoluted tubule is attached to it, the distal convoluted tubule is not. If we were to stretch the nephron out straight, we'd have glomerular capsule, PCT for proximal convoluted tubule, nephron loop, DCT for distal convoluted tubule. That's the nephron. So really the distal convoluted tubule is far from the glomerulus as far as the flow of the filtrate is concerned, but in fact it is technically right next to it, which we actually see in this model here. So in this model we have the arteriole coming into the glomerulus and the arteriole coming out. They have special names. The one going in is called the afferent arteriole, which just means going into, and the one coming out is the efferent arteriole, which again just means going out. And then we have the capillaries here. In the model, the capillaries are shown exposed on one side and then covered in another type of cell on the other side. In fact, they're all covered with this other type of cell, but they only represent the picture on one side. These cells that covered are called podocytes. They wrap around the capillary of the glomerulus, and between the glomerular capillary wall, its basement membrane, and these cells called podocytes, if I didn't say that before, those layers make up the filtration membrane that all of the waste products from the blood come through. And then that filtrate collects in the glomerular capsule, the space outside the glomerulus. Here we see the proximal convoluted tubule coming off of that. That's where the filtrate goes. The convoluted tubule's got bends and twists in it, and then we have the nephron loop going down and back up. And then this is exactly what it is unclear, and different sources will say differently. It's either the very end of the ascending loop of the nephron loop, ascending limb of the nephron loop, or it's the very beginning of the distal convoluted tubule. Which it is isn't terribly important. It's just at the end of the loop. It's right next to the glomerulus, but it's not connected to the glomerulus in terms of the flow of the fluid. Okay? The fluid going through here is not going to get back in the glomerulus. The cells in the wall of this tubule, collectively called the macula densa, can sense the presence of filtrates, mostly sodium and chloride. When the concentrations of sodium and chloride in the filtrate going into the distal convoluted tubule are not precisely what they're supposed to be, the macula densa can interact with structures in the glomerulus, called the juxtaglomerular apparatus. I hate the words that are used in renal physiology, which just means next to the glomerulus. And the glomerulus will change the flow rate of blood through the glomerulus and filtrate out of the blood to get the filtration back to where it's supposed to be. The book goes into a lot of detail about how that's regulated. If I have time today, which I really doubt, I'll talk about it a little bit, but I don't think I'll have time to. There's a series of hormones that play a role that start with a hormone that comes from this part of the kidney, and then it has effects systemically, and the book goes through that. Now, in the middle portion here, the pie-shaped part of this model, they actually represent two nephrons, and that's because there's two different types of nephrons. The book doesn't make quite as much a point of this as I would hope, but I do want to point this out because there's an important point to this. The nephron that we see with a longer loop going deeper into the medullary pyramid, which, though it's not actually to scale in this model, would actually go pretty much all the way to the bottom of the medullary pyramid, almost to the papilla. The ones with a longer nephron loop are called juxtamedullary nephrons. Okay? Another example of why I hate the words used in this. Juxtamedullary means next to the medulla. Okay? And what that's referring to is that the glomerulus is at the bottom of the cortex. It's right next to the border with the glomerulus. Border with the medulla. So this is a juxtamedullary nephron. The other one that we see over here represented has a shorter loop that does not go as far into the medullary pyramid. This is referred to as a cortical nephron. really, juxtamedullary nephrons are the ones that are right at the border. So these two over here and these two over here are juxtamedullary nephrons. All of the rest are cortical nephrons. Again, these models are not too scaled. So they're just showing glomeruli, which is plural for glomerulus, stacked about 3 deep in the cortex. Over here, we see them stacked about 8 or 9 deep. So again, just the first ones that are right next to the medulla are the juxtamedullary nephrons, and all of the rest are cortical nephrons. The capillaries that we see going down into the medulla here that look very much like a loop would be associated with the juxtamedullary nephron loop. A cortical nephron loop does not have this kind of capillary structure associated with it, just those really long loops. Those really long loops with the capillaries that go around them, represented over here on this model, together are specialized for concentrating urine. And the book goes into talking about concentrating urine. Last week when I was showing you some stuff from the book, I said there's that part about the countercurrent multiplier system. That's getting at how it actually concentrates the urine as much as it can, along with antidiuretic hormones roll with that. But the nephrons that have the really long loops are the ones that are specialized for doing that. The ones with the short loops just do your everyday kind of filtration, which is most of what happens in your kidneys. And the book really doesn't make a big point about the 2 types of nephrons, but I want you to be aware of that. So that's the structure of the kidneys, the stuff that you need to be aware of to understand where your analysis goes. Kidneys are just filtering blood. It actually happens in kind of a 3-step process, as I see it. And this isn't something that I made up on myself. I got it from other sources. The current book doesn't quite put it this way, although it does address all of these things in the book. It starts with filtration, which is what happens in the glomerulus. Everything gets filtered out of the blood that can get filtered out of the blood. The filtration membrane, which is the glomerular capillary wall, the podocytes, and the intervening basilar membrane between them, will allow any kind of small molecule to pass through, with the exception maybe of certain small lipids. No large molecules, meaning plasma proteins, and no blood cells can pass through, just small molecules. Now, small molecules includes water. Water is a small molecule. So there's a solution, the filtrate that comes out, has a lot of water. All of that water is being pulled out of the plasma. Also, any small molecules dissolved in the plasma will be pulled in. A lot of those are waste products, which is the whole point behind this. But it also is going to pull things out of the blood that we want in the blood, like glucose, and amino acids, and other nutrient molecules like that. So the second step after filtration is reabsorption. Reabsorption is when things move from the filtrate back into the blood. I showed you a picture last time of the sodium concentration gradient that's set up to pull things like glucose and amino acids and stuff out of the filtrate. That's reabsorption. It'll also pull other small molecules and small particles like ions across back into that. A lot of waste products do not get reabsorbed because there's not a specific mechanism for them to be reabsorbed. If any of you took an aspirin-like drug this morning, Advil, Motrin, whatever it might be, that molecule is in your bloodstream right now. Your liver has probably converted it to an inactive form or something like that, detoxifying the blood. But it's still in your blood, so it has to get filtered out. You don't have any mechanism in your kidneys to move that from the filtrate into the blood, so it stays in the filtrate. But glucose, amino acids, electrolytes like sodium ions and those sorts of things, they will move back into the blood, okay, in reabsorption. because a lot of those things we want to keep in the blood. The third step is referred to in a few different ways depending on the source. I like to call it tubular excretion, which describes excreting things into the tubules. So the blood, as it passes by all the tubules and the great tubular capillaries, some of which extend down into the medulla, will move things out of the blood back into the filtrate. And those are things that we know we want to get rid of. Okay? Actually, very few waste products move that way. Most of the waste products move through the filtration step. But when there are some things that we want to get rid of, under certain conditions, they get sent back into the filtrate during tubular excretion. So if we want to get rid of excess calcium or potassium or sodium or whatever in our blood, that happens in excretion. If we don't want to, we keep that stuff in our blood, okay, at that third step. So it's 3 steps: filtration, reabsorption, tubular excretion. It might seem like, well, why don't we just start with the third step if that's getting things out of the blood? But it's only getting things out of the blood that we know we want to get rid of. Okay? Anything that's in your system that you are not molecularly aware of. You might be cognitively aware of taking aspirin this morning or something like that. But at the molecular level, at the physiological level, you don't have any awareness of that as a waste product in your blood. So we just filter all that stuff out and it stays in the filtrate and you get rid of it. That's essentially how the kidney does its job. Urinalysis gives us a way to look at that, the results of that process. That process is very predictable. So what we see at the end result, what we see in the urinalysis, is a very good picture of what's happening in the blood. Okay? We can assume that certain things should be or shouldn't be in the blood, in the urine, and therefore we can kind of look at the blood and figure out what's going on there. If you see too much of something in the urine, then there's probably too much of it in the blood. You don't see enough of something in the urine, then there's probably not enough of it in the blood. Okay. Now, sometimes the differences between the analyzed urine and what you expect might represent problems in kidney function, and the blood might be fine, but still it tells us something about what's going on. And unless there's other information about kidneys having problems functioning, you can assume the kidneys are working fine, and it's a picture of what's going on in the blood. So that's what urinalysis basically is. Now, you have all done urinalysis before. It's part of the human condition. We're all curious about what comes out of our kidneys. Okay, whether that means you get up and look in the bowl behind you after you're done getting rid of that waste, whatever, it doesn't really matter. But that's urinalysis. It's just looking at the urine and finding out what's going on. The tools you use to look at the urine can get more and more complex, but it can be just as simple as using your eyes. So you can look at a sample and just say, oh, okay, that's what color I would expect it to be. Okay. It should be, and here's what's on the board that kind of corresponds with it, it should be a pale yellow to a deep amber, which I would call a dark yellow. If it is not in that color range, then that tells you that something's wrong. Okay. Now, it says pale yellow, but really it can be clear if you're just drinking so much water that you're passing water at a regular rate and no waste products get filtered out to be visible in there, then that would be the case. But generally speaking, under normal function, it should be somewhere in that range. If it's not that color, then there's probably something going on. I had a student who was very excited about urinalysis one semester because she'd just been prescribed a drug and she was told that it was going to make her urine turn a neon blue color. So she was very excited. She got her cup. She went across the hall. She got her sample. She came back in here, very dejected looking, put it in the back and said, it's green. But why was it green? Yeah, yellow and blue makes green. It would have been neon blue if she didn't have very dark urine. But the fact that she had some yellow color in the urine, it diluted the blue down and became green. So it could be a different color. Also, it should be clear. But it should be clear, which I usually mean to say, you can hold up something behind your sample and you should be able to read through it. If it isn't clear, then there's something else in the urine. Can anybody tell me what the most likely thing that's going to make urine cloudy is? Anybody know? Sorry?
We lost an infection.
Infection. Because of what? What is
Pus.
the sign of infection? Pus, white blood cells. So if we were to take some pus and pour it in to a sample, it would get cloudy. Okay. I always kind of wanted to try this. So I have here a chai latte, which is not exactly pus color, but it's a little bit okay. And so I just wonder what happens if I pour a little bit of that into the sample. Yeah, so we got that. All right, it's lovely.
Yeah.
Okay.
What was that you put in there?
It's my chai.
Oh.
It's a
Yeah, he's
yeah. I carry pus around in my the
Where'd you
places
get
you'll go.
Cheers.
Yeah.
Oh,
No, that's my chai. Anyways, so that's what it could look like if it's cloudy. Okay, that would be a sign of infection. There are other things that can make it cloudy, and I just want to warn people about one thing, which is not actually anything that's a problem. If when you collected your sample, it was nice and clear, but you go to get it afterwards and it's not, don't worry. I was not aware of this until one of my students came up in that sort of situation saying, mine's really cloudy, I don't know what's going on. And I had no idea what it was, so I was quietly freaking out and just said, maybe you should see your doctor and ask if they could do a more formal urinalysis. After class, I looked it up, and it turns out it's not a big deal. There is genetic variation within the human population, if you weren't aware of that. And so there are some people that carry a gene that produces a molecule, a breakdown product of some metabolic reaction, that at room temperature will precipitate out of urine. Okay. So in your body, if you are a person that has this happen in your sample, in your body, it's not doing that. At body temperature, it stays in solution. The fact that I spent all this time after you've taken your sample, it's been sitting in the back. If you have this particular genetic difference, you will have a cloudy sample. Actually, it's kind of weird. It gets really thick too. So it'll really freak the hell out of you.
Yeah.
If it was clear when you got it and it's cloudy now, don't worry. That's just a matter of the temperature change. And that's not happening in your body because your body is not room temperature. So don't freak out about that. But those are the sorts of things that can make a sample cloudy like that. Now the next thing up there is odor. It says odorless. So
Wait, that's got pus in it now.
no, it's got chai. So I wouldn't say it's exactly odorless. Samples
Yeah, I was
tend to have a
gonna
little
say.
bit of
Isn't it got like that, because when you smell urine, you can
Right.
stop.
Yeah. You can
Yeah, and that depends on how much stuff is in there. I would say odorless there is a
Yeah,
little bit of
so-
an understatement.
Yeah.
Okay.
But when there's like an infection, that smell is different than the smell of just regular
Right,
urine.
there's gonna
Yeah.
be particular smells associated with things that might be in there. Probably the best known odor change in urine is not at all a medical condition, it's just the fact of having eaten asparagus. Again, there's a genetic variation in the human population. Some people, when they eat asparagus, produce a very odorous molecule in their urine. What it is the amino acid asparagine, which it's no coincidence that asparagus and asparagine are kind of the same word. There's a lot of asparagine in asparagus, and in fact, the name asparagine comes from the fact that it was found in high levels in asparagus. So some people metabolize asparagine differently than other people, and it makes an odorous molecule that ends up in the urine. Completely innocuous, no problem, other than people with this condition might eat asparagus and be reminded of what's going to smell like in the bathroom in just a little while. And so they can't eat asparagus. But so that's an odor that's associated. A more clinically relevant odor that might be associated with what would happen, what would it suggest if you smell the sample and it smells sweet. How do you sense sugar? We did senses at the beginning of the semester.
Oh.
What interacts with sugar in your sensorium? Sweet is what kind of stimulus? Taste stimulus, which means that the glucose receptors are located where? On your tongue. Right? So if you're smelling something that's sweet, you're not smelling sugar. What you're smelling when something smells sweet are ketones. Ketones are metabolic products of glucose, or not glucose, but carbohydrate metabolism that build up when people have certain metabolic disorders. Diabetes might be one of them, but more likely it's some sort of ketonuria. If you've ever looked at warning label on anything that's got NutraSweet in it, like Diet Coke, it'll say PKU danger or something like that. PKU is a condition called phenylketonuria. And this is another genetic difference, but this one's a bit more serious than the asparagus thing. Those people cannot break down phenylalanine, which is another amino acid, correctly. And so it gets broken down incorrectly, producing a lot of ketones. Those will build up in the system and they can be toxic at high levels. So people that have that condition have to be sure that they don't take in too much phenylalanine. And phenylalanine is one of the components of NutraSweet. So NutraSweet is off limits for people with that condition. You can also see a buildup of ketones for people that are doing a low-carb diet. Okay? If you're not taking in carbohydrates, mainly glucose, then you're going to start metabolizing carbohydrates in your system, which are probably not glucose. They're carbohydrate related, but they might be ribose from nucleic acids. They might be pyruvate, which comes from glucose metabolism. If you have that around, you can break it down a little bit further. But those types of metabolic reactions produce more ketones. Now, usually if you're eating a low-carb diet, you're not producing ketones at the level that it's going to be toxic. However, if you're on a low-carb diet and you're in, what's that called? Ketosis, which means that you have a high level of ketones in your blood, you can smell that on the person's breath, okay? And you'll smell it in their urine. Saliva is just a filtrate of the blood also. So if there are sweet-smelling ketones in the blood, they'll be on the breath because they're in the saliva. Okay? And you can kind of tell that. If somebody has very sweet-smelling breath and they don't look like they're on the South Beach diet, then there's something else going on, and it's probably going to be toxic to the blood. But that's what smelling something sweet would be. Okay? Now, urinalysis is something that's been done for a very long time. There's evidence actually of urinalysis being used to diagnose somebody with diabetes back in ancient Egypt. Now, that statement has a lot of stuff to unpack. The term diagnosis is not an Egyptian word, it's a Greek word. So the Egyptians didn't have a concept of diagnosis in a medical sense necessarily. And then diabetes is the name for this disorder in the modern age. So certainly back in the ancient times, they didn't call it diabetes. But they knew that there were certain people that tended to have problems and they'd lose feeling in their extremities to the point possibly where they'd start seeing damage to their fingers and toes, and people would die eventually of problems related to that or something like that. And they realized that people that had a high level of glucose in their urine were the ones that are going to have that problem. So the relationship of glucose in urine to diabetes, whatever it was called back then, was known. Okay. How could somebody in ancient Egypt know if there are high levels of glucose in the urine? I've already set up the answer to this question. Yeah, you got glucose receptors on your tongue.
Oh, own apple.
Okay, that's the first time I've done that with the child. But that was in fact not a urine sample for everybody's benefit. That was green tea. I discovered several years ago that green tea is a perfect substitute for urine sample. I think the thing that really sells it, notice there's a little condensation in here. That's because it's hot tea. But yeah. And actually the chai green tea thing kind of works. So Chelsea?
I was just wondering, I know you can test someone's blood and their urine, but I saw that at the hospital workup, they take spit samples and they use that for, it's like a study, people who have a diagnosis of diabetes.
Mhm.
And I don't know, how does that work? What is that?
Well, so spit, saliva, and urine are just filtrates of blood. So if you look at either of them, you're going to be able to see something about what's going on. It's just that the physiology of urine production is a little bit more tightly controlled. So it's probably a better place to get a broad idea of what's going on. But for something specific, if it's like diabetes, there might be certain markers that they're looking for that they know would be present in saliva. But it's not a general.
You wouldn't dip it like you would urine, is that correct?
I don't know how to test it, but yeah, dipsticks are usually color reactions. Usually the best way to do this kind of test. So we'll talk about that. So back to me drinking the pee.
That
So-
freaks me out.
Oh, good. This is my favorite day of the semester. So I did not do it just to gross you out. I also did it to kind of reinforce the idea that urinalysis is just anything you can do to figure out what's going on in the sample. Okay. Now, luckily, you're going to be doing it in a much more modern way, but I could, sight, odor, taste, all of those are ways to find out what's going on in urinalysis. And everybody's done that to some extent, whether it's formally or just curiosity of what's in the toilet bowl behind you or something like that. It's just a very easy thing to do. But what we're going to do is a little bit more involved. And there's two tests or two procedures we're going to do. And I just realized I should have set some of this up earlier. The first thing we're going to do is a multi-test dipstick, which I'll show you in a second. So in this bottle here are strips of little colored squares. And each of these squares has been pre-treated with some chemistry which will react with the sample, and the color reaction coming from that will give you an indication of what's going on. Okay. So all of the squares on here are represented in this graph on the bottle, and after you've done the reaction, you can hold the strip up next to the colored squares on the bottle and see what kind of reaction is taking place. And you can just, for each square, kind of slide it across and compare the colors until you find the color that matches closest to what's on the bottom. Now, every so often we get one of these, and it's usually the urovalinogen one, that comes out some weird color that's not on the bottom. If that's the case, then the test just didn't work. I think that particular test is very sensitive to oxygen, so if it's exposed to oxygen in the air around us, the color reaction stops working. And that usually happens because people leave the lid off of these bottles too much. We have one bottle that has some sticks in it. And then we have 3 more bottles that don't. Okay, these are empty. We have these, however, so that you can compare your stick to those as well as this one here. We have 4 bottles for all of you to work with, so there will have to be some sharing, but at least with 4 bottles instead of 1, you can make the rounds a little better. When you're coming up to get the stuff, which you'll do in a little while, get your dipstick and get a bottle if it's available, or be around somebody that has a bottle so that you can look on it. The other test that we're going to do is sedimentation. Oh. Uh, Sedimentation means that we are going to spin the samples down in the centrifuge and separate the solids out from the liquid. And then we'll remove the liquid and put the solids on a slide to look at in a microscope. This poster here shows examples of the kind of things that you see in urine. Okay. And I'll talk about this when we get to that step. So let me explain how this is going to work. Now I'm going to be using my green tea sample here as an example. So remember, this is not actually urine. You'll need a conical tube, which there are a bunch of. Not all of them have a scale printed on them, but you need to put 12 milliliters of sample in your tube. So I'm going to leave my sample tube up here, which again does not actually have urine in it, it's green tea. And if you happen to grab a tube that doesn't have anything printed on it, like this one, then make sure that your tube is filled up the same as mine. They match the height of this. Now this tube I happen to grab actually has a little Sharpie mark on it. Somebody put on there at the 12 milliliter mark. Okay. So you might see that, but I've also seen some here that are marked at the 10 milliliter mark. So make sure that you have the same volume as me. We need them all to be the same volume because when we spin it in the centrifuge, it has to be balanced. So if one has 12 and one has 11 and one has 10, they'll be all off balance and the thing will not work properly. So make sure that yours has this much in there, and if you don't grab one with the marks on it, compare it to mine. If you did not happen to collect enough to get 12 milliliters out of here, after you do the dipstick part, you can add water to bring it up to full volume. Okay, that's fine. So before you do that, however, if you have less than 12 milliliters, put the dipstick in to your sample and get it wet, and then put it off to the side to dry. I would suggest you grab your own paper towel, put it there to dry. On the bottle with the dipstick thing here, for each of the tests, it says how long the test needs to react. So glucose, which is the first one, needs 30 seconds to react. Nitrite, the second to last one, needs 60 seconds to react. The top one, leukocytes, needs 2 minutes, so just one, but people are not likely to see leukocytes. So you need this to sit at least a minute or so. If it sits a little bit longer, that's fine, but it needs at least that long for the reactions to take place. And then after it's reacted, just hold that strip up next to the scale on the jar and you can read it. So for instance, the first one is glucose, and you would expect in a normal sample for it to be this kind of pale teal color. Mine, of course, is green tea that's been sweetened with Splenda, and I added the chai to it. So it's going to register as very full of glucose because it's been sweetened. And for some reason, I've done this without the chai, and it still reacts like this. Splenda, since it's chemically so close to glucose, reacts with this similar. So you can just compare each color reaction across the bottle. So after you've gotten that sample from your, the dipstick in your undiluted sample, if it isn't 12 milliliters, bring it up to 12 milliliters with water so that it will balance. And then everybody takes their tube back here, and I'll spin it around. The centrifuge has 8 spots for spinning tubes of this size, and there's definitely more than 8 each, so I'll have to do a couple of spins. But I'll hold this up in front of you, and I'll point at whichever of the 2 positions in the carrier that you go in. There's a number right here on the front, so I'll say you're number 1 or you're number 2. I will try to remember to say number 1 of the first spin, number 1 of the second spin, that kind of thing. But pay attention. Okay, when I'm putting it in. And if you give me your sample after I filled up the first spin, I have plenty of these, so I'll just take yours and put it up the side to keep the second spin. There will be a little break while this is spinning. What I'd like you to do while it's spinning is to get the microscopes out, which will not take you as long as a spin takes, but go ahead and get the scope out. And then you'll have some time to kill. So if you'd like to take a little bit of break, I know none of you have to go to the bathroom now, but if you want to take a little bit of break while it's spinning, that's fine. Just get the microscope out so it's ready. Don't be gone too long because I want to explain how things happen with the preparing the slides afterwards. But that will count for today's break. Okay, so don't take 20 minutes. We don't really have that much time to take, but feel free to slip out for a little bit. If you have to slip out to eat a snack or satisfy a nicotine habit or whatever it might be, just go out into the hall to eat, and I'll let you know, and I need people to come back in. And 2, don't smoke; it kills you. So that could be the break. So go ahead and get the tubes and the dipsticks and your samples. Oh, I forgot to say this. Don't get rid of your cup just yet. We need that for cleanup later on, which I'll explain after sedimentation. So just do what I tell you so far.
Oh, man.
If you feel that you need to put on gloves, feel free, but really it's not necessary if you're only handling your own sample.
It's so secure with that, right?